Mesdames, Messieurs, bienvenue à cette conférence de ce soir du professeur David Lee. En fait, on va avoir droit à... Vous avez le titre ici, donc vous verrez un petit peu de quoi il retourne. Mais juste pour le présenter, je voudrais quand même expliquer que ça fait déjà des siècles, des millénaires que l'homme utilise l'énergie musculaire. D'abord sa propre énergie musculaire, puis celle des animaux domestiqués, et puis, depuis pas si longtemps, finalement, dans l'histoire de l'humanité, on utilise des machines de plus en plus sophistiquées, des machines complexes, des machines énormes qui peuvent soulever des tonnes, des machines volantes. Et puis, qu'est-ce que c'est qu'une machine? C'est quelque chose qui provoque un mouvement contrôlé. Or, je bouge, j'ai des mouvements contrôlés, tous les humains bougent, tous les êtres vivants bougent, la vie bouge. Comment est-ce que la vie provoque des mouvements contrôlés? Eh bien, ça se passe au niveau cellulaire, ça se passe au niveau moléculaire, et par conséquent, la nature a trouvé des systèmes moléculaires capables de bouger, des espèces de machines moléculaires qui vont pouvoir provoquer un mouvement, le battement de cœur, marche, etc. Et le professeur David Lee a une passion, c'est celle d'essayer de reproduire à l'échelle moléculaire de telles machines. Non pas imiter ce que fait le vivant, mais utiliser la chimie qui est une science d'invention, qui crée son objet, une science qui permet de créer de nouvelles molécules qui pourraient avoir le même type de comportement. Et c'est ça qu'il va nous présenter aujourd'hui, mais brièvement, je voudrais présenter un petit peu sa carrière. Il a fait ses études à Manchester, puis un postdoc au Canada, puis il est revenu en Angleterre, à Manchester de nouveau, puis il a eu un poste de professeur à Édimbourg, et on apprend récemment qu'aujourd'hui il retourne dans son alma mater à Manchester. Professeur David Lee a reçu beaucoup de prix, pas encore le Nobel, ça viendra peut-être, mais entre autres, par exemple, le prix Feynman de nanotechnologie, le prix Descartes, et puis il a été récemment aussi élu à l'Académie royale anglaise. Je ne voudrais pas terminer sans dire que David Lee est aussi membre du cercle de magiciens d'Édimbourg. Ah, je crois que vous allez voir des choses intéressantes. La magie moléculaire. David Solios.
Okay. Thank you very much, Alex, for the kind introduction, and my thanks to the Wright Foundation and the Wright family for making it possible to bring us all over here this week for this wonderful series of colloquia that we, the scientists, have really enjoyed. And thanks also to the people of Geneva for making us so welcome and making sure that the week that we've had has been so much fun for us scientists. Right, the theme of this colloquium series is molecular architectures. And the molecular architectures that I'm going to talk about today are all based on these sorts of things, rings. And we'll pass this around. So if you could pass this around and back, we'll get back to it later. Because rings have got a really important place in molecular architecture, but you have to have the right sort of rings. If the rings are too big, then lots of different things can go in and outside of those rings, and you lose some of the benefits of that molecular shape. If the rings are too small, then nothing will fit inside the rings, and again, you lose some of the benefit of the cyclic shape and topology. But if you choose the right sort of rings, which have got the right size of hole in them and the right sort of chemical structure, then you can bind things, other molecules, selectively with inside the cavities of those rings. And if you choose your rings carefully and you've got the right sort of cyclic structures and they're the right sort of size and shape, then if you're very, very careful and very, very lucky, then strange things can happen. Strange things can happen. No, no, no. No, 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 no. No. But I am available for birthdays and bar mitzvahs and Christmas. But really what I'm going to talk to you about is the sorts of surprising things you can do with these rings, the architectures that you can create, and how you can use these sorts of cyclic structures to create molecular-sized machines. So really the size of these structures, to give you some sort of idea about that, we're really talking about the smallest we can possibly make a machine. So 50 years ago, a computer would have filled the size of this stage. And we know that in the subsequent 50 years, now the computer chips that are found in your iPhones and smart devices, the actual chips can fit on the size of a pinhead to have the same calculating power as something that would fit in a machine. But I'm not talking about going in terms of scale down from meters to sort of millimeters. What I'm talking about is going right the way down to the bottom, to things that are a millionth of the size of these sorts of machines, down to the nanoscale, or things that are a millionth of a millimeter wide. And there are high hopes for these sorts of machines. People hope that they're going to create a sort of revolutionary nanotechnology which will allow them to do all sorts of amazing things, both in disease and in other sorts of areas. But there are fears as well. And some people, such as the Prince of Wales in my country, that very eminent scientist, is really concerned that there's going to be self-replicating nanobots that are going to take over the planet. So what's the truth of these things? What's the science fact and what's the science fiction? Well, in this sort of research area, what we try and do in our group and in our field is to follow the advice of Alan Kay, who was an American computer scientist who worked for Hewlett-Packard and Apple. And at the times that there were all rumors going around in the 70s of 80s, oh, Apple are going to do this, oh, IBM are going to do that. Alan Kay cut through to the heart of the essence of the issue of how to predict the future by saying the best way to predict the future is, in fact, to invent it. And as scientists, we're able to do that in the field of molecular machines by using synthetic chemistry. And chemistry is sort of unique amongst the sciences in that it allows you not only to study how things are in terms of biological machines and biological structures, as we've heard this week and will do tomorrow, but also to create new things, create things that haven't been made before, and to invent new types of systems that can have exciting functions. And that's the field of molecular machines. It's going to be what we choose to invent it to be. But why do we think that it's going to be possible to shrink the concept of a machine down to the molecular level anyway? Well, the answer is that there already is a working nanotechnology out there. It's the one that's used by nature. And this slide actually shows the reason why scientists should be perhaps trying to make molecular-sized machines. And it's because biology, nature, uses molecular machines for everything. So the way that you're able to look at the board, the way that your muscles move, the way that energy is harvested from the sun, the way that replication occurs, DNA replication, the way that information is stored in the cells. Every single biological process depends fundamentally on molecular machines that control molecular movement. And in contrast, at the start of the 21st century, mankind doesn't use molecular machines or control molecular motion for anything at all. So every pharmaceutical, every drug, every chemical reagent, every catalyst, every material in our technological world just depends upon the static or equilibrium properties of the molecules. And biology hasn't evolved over 2.5 billion years to use molecular machines for everything for no good reason. And when scientists learn how to actually put molecules together and control their movement and allow them to interface with other molecules, then I'm sure it's going to revolutionize our complete way of approaching functional molecule and material design. But the way that we're going to do that is not to shrink down motor cars to the molecular level, we can't do that because, unfortunately, the way that matter behaves at different length scales is just so different. So what we have to do is to try and learn from biological machines, see how molecular machines are made in biology, and then use those design principles to actually design our own sort of structures. So, for example, to show this sort of effect of scale, this nameplate is just going to stay there on the lectern unless I give it some kinetic energy, because it's a big object in the big world. But at the molecular level, things aren't stationary, they're constantly moving. And you don't have to give them energy in order to cause them to move, to make the components of machines move. They're already moving at the molecular level. What you have to do is to actually control that movement, get rid of the random movements that you don't want, and what you're left with is how to actually make machines. And the way that biology does that, and the way that we're going to copy it, is to use architectures which restrict the degrees of freedom of the components. So if we look at these sorts of structures, we'll see that many of these machines move along tracks or rotate around axes. And we're going to make exactly the same sort of architectures with for our sort of machines. And in fact, these are the sorts of architectures that we're going to use. This sophisticated molecular model is supposed to describe one of those sort of molecules where each of these red and blue rings is sort of a molecular structure. And these can't be broken apart without breaking one of the rings, but they're free to move with respect to each other. And these sorts of molecules are called catenanes, from the Latin catena, meaning chain, because these are a bit like links of a chain. And what you can do with a catenane is perhaps control the motion, the rotation of one of the rings through the other, and maybe use that as the basis for a controlled motion in a molecular machine. The second sort of architecture that we're going to meet is where one of those rings is replaced by a linear axle. And the ring is able to move along the axle, but it's stopped from coming off by these bulky stoppers. And this sort of architecture is called a rotaxane, from the Latin rota, meaning wheel, and axis, meaning axis. And again, the nice thing about this is that you can control the translational motion, and if you can control translational motion and rotational motion, then that's everything. But how are we going to make these sorts of molecules? It's hard enough to thread the eye of a needle in the big world. How are we going to thread the eye of a needle when the needle's eye is only 1 nanometer, 1/1,000,000th of a millimeter across? Well, the answer, again, is to use an idea from nature and use this concept of self-assembly. And I wanted to try and demonstrate for you sort of how self-assembly works. And I was trying to think of how to do that. And then today, I went down to in the hotel to get the newspaper, and you can see that the landlady has a dog and had got to the newspaper before me. But it got me thinking that, of course, a newspaper is a very complicated information-rich structure that requires lots of effort from lots of men and women to put together every day. You need your journalists to write the stories. You need your copy editors to edit them. You need the page designers to lay out the pages. You need them to then be printed and distributed. And it's a really complicated way of putting together such a big structure. And I always wondered, wouldn't it be sort of easier if you could take the sort of simple building blocks and get them to sort of self-assemble to and get them to self-assemble to build your sort of structure yourself. And that's the sort of things that we try and do in our group, try and get the building blocks, the molecules, to make themselves. But in fact, we got into this area by chance. We weren't deliberately trying to make these molecules. Some 17 years ago now, we were trying to make molecules that look like this sort of ring-shaped structure. which is designed, it's the right size and shape to bind to carbon dioxide. And we wondered whether we could use this to maybe scrub carbon dioxide from the atmosphere. But when we tried to make this molecule by mixing together these commercially available chemicals on the left, we got a surprise because we didn't get this one-ring molecule at all. What we actually got were two of these rings interlocked together to form a catenane, this cyclic structure. And it's perhaps easier to see from this structure of how the atoms appear in the crystal. One of these rings is shown in yellow and the other is shown in light blue. And the actual reason that this forms is that these dotted lines represent hydrogen bonds, which are the same sort of things that hold water together that we heard of Takoso Aida earlier this week. And these actually template the formation, stick together the two rings, and cause this threading reaction to occur to form the catenane. And this is a one-step, 8-molecule condensation, 4 of these molecules and 4 of these, to form a catenane. And it was, when it was discovered 17 years ago, and still is today, one of the simplest ways of forming these threaded structures that's been discovered. So this was just an act of serendipity, a surprising thing that happened in the lab. But surprising things happen all the time. Surprising things happen all the time. And it's not just have something surprising happen that's the important thing. Of course, it's what you recognize that you can do with it. So we discovered this strange way of being able very efficiently to make these interlock rings that can undergo these large motions. We wondered, could we use that to actually control the motion of these systems and actually use it to use it as the basis sort of molecular machines. And so for what I'm going to try and tell you about today is about how you're going about doing that. So it turns out you can make not just rotaxanes this way, but also, not just catenanes this way, but also rotaxanes. So who has the ring that we just passed out? Does someone have that in the audience? Who has the metal? Oh, could you come down and give me a hand, sir? Would you mind? If you don't mind, if you could just come up on the stage round here so you don't fall off. If you just come up, and then we'll come round here. No, quick. Dépêche-toi. And so I'm David. Je m'appelle David. Simon, nice to meet you, Simon. Okay, we're going to try and do some chemistry now. Okay, a demonstration. If you hold out your ring like this, that's great, fantastic. So we have the ring, we have the thread here, we have 2 bulky stoppers, one much bulkier than the other, and what we'll try and do is form the rotaxane. So I just need to put a bit of catalyst on here. There we go. Right, and if you just drop it, Simon, just drop. Bravo. Well done. Fantastic, Simon. Well done. Big hand for Simon. Well done. And I'm pleased to say it works just as well in the lab. You see 100% yield, just as Simon got. So what actually was happening there was that in the thread there was a carbon-carbon double bond, and this catalyst that I added opens and closes this double bond many times, allows the ring to thread on, and you can get these really great yields. So we can really make these sorts of molecules, and it turns out, sorry for all the chemical structures on this slide, but this is just to show that this reaction's actually very structurally tolerant. You can make lots and lots of different types of molecules, threaded structures, using this chemical reaction. Some of them work really well, and some of them involve biological substrates, and so on. So it's really possible to make these sort of things. What can we do with them? Well, to make a molecular machine that exploits these sort of interactions, one way to do that is to have 2 different binding sites for the ring on the thread. And if we have that, then if you remember I said that all the components of molecules are moving at the molecular level, and so in fact the rings actually move between these 2 sites, but they'll prefer to stay on whichever site is the stickiest for the ring. But because we're chemists, we can do chemistry on this molecule and perhaps change the chemical structure of this binding site, make it chemically different and perhaps not so sticky. And if we do that, then all these Cheerios are going to go to this end, and the net position of the ring on the thread will change. And if this is a reversible reaction that we can make go the other way too, then what we have is a mechanical switch that works on the molecular level that's reversible, that we can move back and forth. And we can use lots and lots of different stimuli, lots of different sort of forces to make this switching effect work. We can just change the polarity of the environment. So we can change it between being oily and water-like, and use that to bring about changes of position of the ring on the thread. Or we can, if we design our molecule differently, we can use light or electricity or temperature or binding to various different sorts of anions. All of these things, if you design them properly, you can use to bring about these switching events. So what can you do with that? Now we can make the molecules, we can move the parts, but what can they do? Well, it's really just up to the imagination of the scientist, because we can add bits to the ring and to part of the thread-like structure, and in one state these will be held close together, and then if you drag the ring away, you'll pull these bits apart and you'll switch off whatever interaction that you designed in for them to have, and then you'll be changing the property effects of the molecule by controlled molecular motion, by machine-like action. And here's an example of the sort of thing that you can do with this, which goes back to Professor Holmes's lecture yesterday, where he was talking about fluorescence and showing these fantastic examples of fluorescent polymers. So this is a fluorescent group on the end of one of these very simple molecular machines, and it's fluorescent. So as Andy showed, you shine UV light on it and it gives out light of another color, this beautiful blue anthracene fluorescence. But on this ring on the thread, we have a group that can quench the anthracene fluorescence, so basically stop it being able to emit light. But it only does it when the ring is very close. So if the ring is switched so that it's close to here, we have the molecule in the off state, so you shine light on it and nothing comes out. And if you move the ring away, you get this lovely on-off optical response that you can see with your naked eye just by controlled molecular motion. And this works in polymers. So if you coat these simple molecular machines into polymers, you can write information in the polymer film, in the plastic film, that you can see with your naked eye that's just a result of controlled molecular motion of the ring up and down the thread. We can do other things as well. This is a Teflon-like group on the thread, and the ring can either be switched to be away from the Teflon group, so exposing it to the outside world, or we can move it by shining light on the molecule and causing a chemical change in the shape of this group here. We can cause this ring to move and cover up the Teflon-like group. Now, what can we do with that? Well, we can coat these molecules onto a surface. This is a gold surface, which is just a very, very flat surface. And we can put this single layer of the molecular machine molecules on the surface. And if the Teflon groups, the fluorocarbon groups, are exposed to the outside world, then the surface will be what we call hydrophobic, which is just like the Teflon coating of a pan. But if we shine light on the molecule, we can cause these groups to move, the Teflon groups get covered up, and the surface changes so that it's not phobic anymore. It actually likes watery substances. And this is a picture in movies of what you can do with such materials. So this now is a tiny droplet of a liquid. It's not actually water, it's diethylmethane, because this is a very, very tiny droplet, just a microliter, so just over 1 millimeter wide. And so you have to have a very high boiling point liquid because it has a very high surface area to volume ratio here, so it doesn't evaporate. And this is on this gold surface, which just has a monolayer, a single layer thick of these rotaxane molecules on the surface. And what we're going to do is radiate light on this surface, and you can see there's a large contact angle here because the droplet doesn't like the Teflon groups. And this is a video of what happens when you do that. So this is the droplet on the surface here, which just has a single layer thick of these molecules on the surface, and we're just irradiating at this point, which is covering up then the Teflon groups in this region here. And you can see that then the droplet starts to move along this surface of molecular machines, and then even the rear end is moving. So the whole of this droplet that you can see with your naked eye is being moved by one layer thick of molecules, of machine molecules on a surface, and you can use that to transport an object along a surface. You can If you don't have the Teflon groups, you don't see any change of the in the droplet and no movement, so we know that this is the cause of the effect. You can even use this to do work. So this is a 1 in 8 slope, which is probably more than I can do on a bicycle. But if you put the droplet on this surface, you can drive the droplet uphill against the force of gravity. Remember, this is just a single layer thick of molecules on this surface, and you can drive a droplet that you can see with your eye up a hill against the force of gravity by the action of light on this surface. And these things are really getting through, generating a lot of force. So this distance that these little units are moving is about a nanometer, so a millionth of a millimeter. And so in terms of effective scale, this is like having a load of pistons at the bottom of the CN tower, and them all moving just 1 millimeter and being able to raise an object more than twice the height of the CN tower. So it's an enormous distance that these things are propelling the object uphill in terms of effective scale. But those things really, the things I've talked about so far, are just sort of switches. How can we make things that are a bit more like biological motors, such as this molecule that walks along a microtubule? It's called kinesin, or this amazing molecule that we're going to see more of tomorrow, the ribosome. How do we make motor systems? And the answer is that, in fact, chemists need to use solutions that are already known out there, but in a different branch of science with a nasty-sounding name of non-equilibrium statistical physics. And really, what does that mean? Well, this story goes back actually 150 years to the time that the laws of physics, of thermodynamics, had just been formulated by Lord Kelvin and a scientist called James Clerk Maxwell in Scotland and Boltzmann in Germany had come up with the theory of heat, which said that, in fact, gases were made of molecular structures. And it wasn't believed by lots of people, but people like Maxwell and Kelvin and Boltzmann did believe that. But these foundation of these laws brought up lots of paradoxes that the scientists couldn't explain. And one of these is sort of articulated in a thought experiment, an imaginary experiment that Maxwell came up with to illustrate this sort of paradox. So Maxwell thought of, what about if I had a box that had some gas molecules in, and the red ones are fast-moving, so they're hot, and the blue ones are slow-moving, so they're cold. And imagine that there's a barrier between these two boxes with a trap door. And then every time a fast-moving molecule approaches from the left, some imaginary being recognizes that, opens the door, and lets the fast-moving molecule move from left to right. And cold molecules, he opens the door to allow them to move from right to left. But he doesn't allow slow-moving ones to move from left to right or fast-moving ones from right to left. In other words, this being that's become known as Maxwell's demon is able to sort the molecules by their temperature or their speed, and in doing so, he could set up a temperature gradient if that was possible to do. But if this was a frictionless door that the being was operating, then he would be setting up this temperature gradient without expending, by the looks of it, any energy if the door didn't require any energy to open or close. And yet, if you set up a temperature gradient, that's just what you have in a piston or a turbine, and you can use it to actually do work. So this would be a way of making a perpetual motion machine, getting energy for nothing, which Maxwell recognized couldn't be possible. It would break some of the laws of thermodynamics that Kelvin had developed, but he couldn't understand why it wasn't possible. And he realized that there were other sorts of demons that would also cause similar sorts of problems. You didn't have to sort according to speed of movement or temperature. You could even just sort according to position and have molecules, doors being opened just to let molecules through from left to right and not from right to left. And this sort of effect would allow a pressure gradient to be built up. And again, it looks like the demon would be expending no energy to do that, and you would create pressure, which again you could use to drive a turbine and get energy out. So how was this possible? Well, it took the best part of 100 years for people to figure out the full rationale for this paradox, and it's in fact to do with the relationship between information and energy. So the information that this being has to process to know when to open the trap door or not is more than the energy that you can ever get back from this. And this is why laptop computers always get hot. You can't do information processing for nothing. But from our point of view of making molecular machines, it's very interesting because what we're able to do in these sorts of systems is somehow the Maxwell system is able to generate directional movement of molecules even when in a system where they're able to go back and forth and undergo random motion. And so this is interesting, this sort of behavior from our point of view of learning how to make motors, because if we can do this sort of thing, even if we're putting energy in, which we'll have to do not to break any laws of thermodynamics, this would be how to make a molecular motor. And a few years ago, we were able to demonstrate this sort of principle. So this is Maxwell's Gedanken experiment, thought experiment, and this is a molecule which does exactly the same sort of thing. It's got two compartments and a ring on this thread, which corresponds to the sort of gas molecules, and this barrier between them. And what we want to do is to set up a system whereby this barrier opens preferentially if the ring approaches from the left. But when it's on the right, the machine somehow realizes that the ring is there and it's not allowed to go back the other way. And if we can do that, then we can actually drive the population of particles, of rings here, from the left to the right without ever changing their energy. And this would be what physicists would call an information ratchet mechanism. It's called a Brownian ratchet mechanism. It's relying on information processing. But this would be doing it in a molecular structure, in a real sort of thing. And so we were lucky enough with some very clever students to actually design such a system that worked. And this is actually the amount of rings on one side of this molecule. And if we shine light on it, we're actually able to drive the populations of the rings over to the other side. This is actually a barrier here, and generate more of the rings on the right-hand side on the left. And so this then is, in a molecular molecular system, an information ratchet mechanism that can be used to power molecular motors. So how can we use that? Well, these are the sorts of molecules that we really want to be inspired by, these biological systems that I hinted at before. This is kinesin, this molecule that walks down polymer threads and carries structures from proteins from the nucleus out of to other parts of the nucleus. And it's a really complicated molecular machine, but how can we mimic these sorts of things? Well, again, I can't emphasize this stuff, that the way to do this is not to try and mimic the sort of motions, smooth motions that we saw before. At the molecular level, movements are much more random than we expect, and this is we're likely to have to make things that go backwards, forwards, take double steps, and so on. And so this is much more like the way that kinesin and the sorts of molecules that we make really move. And so the way to actually make these sorts of structures then is to make things like this, where we have molecules which have got 2 feet and a very simple, just 4-foot-hole track. And what we need to do is to have the feet detached one at a time. This one stays detached while the other one moves, and so on, to allow us to move up and down the track. And if we can do that, we can get repetitive, directional, processive motion. And this is the actual molecule we made that could do that. And it has feet that bind to different sorts of footholds, chemically different footholds. And by playing with these chemical processes, we can actually start by having the ring on the left-hand side and make it take a step at a time and move up and down and walk along the track. However, this molecule doesn't move directionally. All it does is take steps randomly back and forward along the track. And to make it move directionally, which would requires work to be done, we have to introduce a ratchet mechanism. And in this molecule, we do that by adding this unit here, which is something that changes shape when we shine light on it. And if the walking group isn't over the top of it, that really doesn't matter. But if it's part of a ring with this unit in, and it changes shape, it puts a lot of strain energy into the walker, and you can use this strain energy to actually do some work. So to illustrate that, here's the sort of walker with this extentable bit, and this is the one without it that's not actually directional. And I'll try and show how a ratchet mechanism makes this able to move directionally. So we start off with the walker on the left-hand side, and these are all these molecules here. And if we allow this foot to step forward, then what will happen is half of these molecules will say step forward to give you this sort of position. And then what we do is put some strain energy into the structure here. So we put this strain energy in, and this raises the energy of the molecules of the walkers that are in this part. And then when the left-hand foot is allowed to move, far more molecules actually walk to the right than actually go backwards, whereas in this case, just as many go back as actually go forwards. And so that is actually, this is just showing that this really works. And this is actually the first example of a small molecule linear molecular motor. It works by this ratchet mechanism, and we can actually drive it in either direction along the track. So does size matter? My wife is always telling me it does. I say no, no, no. This is actually kinesin. This is our molecule. And kinesin, this big biological walker, takes very large steps for a molecule, 8 nanometer steps, whereas ours takes much smaller steps. And this hydrolyzes a molecule of ATP every time it takes a step, whereas we either shine light or switch between acid and base. Our molecule is already quite possessive. It takes 37 steps on average before it falls off the track, whereas wild-type kinesins typically take about 100 steps. And our molecule can even do something that kinesin can't. It can walk in either direction depending on how we program the reaction conditions, whereas biological proteins always move in one direction. But, of course, the great thing that the biological motor proteins can do is that they can carry cargo and they can move along polymers, and we can't do that yet. Those are the next things that we need to be able to do. But it's still amazing that something that weighs 250,000 Daltons, so a really big molecule, can be mimicked. Some of its properties, in a very, very basic form, can be mimicked by something that's just 21 atoms big. Okay. What about the ultimate of machines that in the universe today, the ribosome? We're going to hear much more about this molecule tomorrow from Venki. But just to say now, this is the most amazing machine that there is. It's what actually makes all the proteins in your body. And it does it by taking messenger RNA, so information from a strand of RNA, and taking tRNA building blocks and putting them together to actually form peptide chains that fold up into proteins. And it's a really incredible machine. How can we make a molecule like this that makes other molecules? This is an extraordinary molecule. It's got a molecular weight of 2.5 million to 4.5 million. So at least 10 times bigger than kinesin, which itself is a big protein. How can we begin to mimic these sort of structures? Well, here is a very, very primitive artificial ribosome based on a rotaxane architecture. So what it contains is a ring with this reactive sort of robotic arm, and a track which has got building blocks on the track which block the movement of the ring along the thread. And what happens is that the idea is that this ring is going to move along the track, and it's got a catalytic arm which is going to pick off the barriers in front of it that are blocking its path, and then it's going to pass those building blocks onto a site here where the building blocks can be assembled to actually make the oligomeric peptide. And once this building block is detached, and the building block transferred to here, the arm is regenerated, and it can move along. So this is the chemistry that we're going to use this is going to be the sort of catalytic group here, and this is going to be the elongation site. And the way this is going to work is through a process called native chemical ligation, and we're going to set up a system where this happens catalytically or iteratively. What's going to happen is this catalytic group is going to come along, detach the building block here, pass it from the catalytic site to the other site, regenerate the catalytic group, and this is then free to move along to the next building block, pick that up, transfer that then to the first building block, regenerate the catalytic site, go along to the final one on this very short strand, break the bond, detach this, attach it to here, and then fall off the track. And so this would be a synthetic molecular machine that can synthesize an oligopeptide, a very short peptide, with a specific sequence that comes from the thread. It actually works in the opposite direction to the ribosome. It synthesizes the amino acids in the opposite direction. So this is actually the chemical structure, much too complicated to go into. But just as this is how we make it, we actually take the strands, build those up, the building blocks on the strands one at a time, assemble the ring around this so that it can't come off the track and go and interact with other molecules, which would stop the sequence. We then add the robotic arm, the catalytic arm, to the top to make the full machine, and then activate it so that it's ready for action. And when we do that, we get a single product out at the end. This single product has got all the chemical groups in it that we expect, so exactly one alanine, exactly one leucine, one phenylalanine, 2 glycines, and a cysteine. And the sequence is exactly the one that we anticipated, because we can look at how this molecule breaks down by mass spectrometry and show that it's got the identical sequence to the ones that we would have from the track. So that is what we're hoping to do with that, is to make longer peptide chains, turns helices, and even maybe mini proteins, use other sort of reactions that biology doesn't do, and eventually use this to make sort of molecular factories and molecular assembly lines. Okay, I've used up way too much of my time. My apologies for that, but I just want to finish by talking to you about one final sort of thing that we met earlier. Because when I was coming over here, I was thinking that, chemistry, it's really a lot like love. That there's a special someone for all of us, a particular one for all of us, but sometimes almost anything will do. And I wanted to sort of illustrate to you how much chemistry is like love. And last night I was sitting next to a very lovely lady who I hope that she'll won't mind coming and helping me with this experiment. So Margie, could you just come down and actually give a hand with this if you don't mind? That would be really kind of you. So big hand for Margie, everyone. So, thanks, Margie. So, Margie, we were talking about love and romance last night, and come up, Margie. And come. And Margie was telling me that she's married to the most wonderful guy, who's actually the person who invited the speakers, or some of the speakers over here today. It's Alan Williams. And I was very surprised by your description of him, because you were saying he's a wonderful man, he's lovely, he's definitely my soulmate, and all these things. And those of you who have met Alan might find it difficult to Alan, just stand up and show the audience what a fine figure of a man Alan is. So okay, thanks, Alan. So I was really touched. That's fine. You can sit down now, Alan. That's your piece done. So I was really touched by that. But I'm a scientist, so I like to see experimental verification of these sort of things. So I want to give Margie the chance, to sort of trade up a bit, really. And so we want to try and find out who is the perfect guy for you, Margie. So we're going to try and do this, but we're going to do it sort of randomly. Okay, so what about Brad Pitt? Is he your sort of... No, not so much. What about Tom? Is Tom Cruise? Absolutely not. Oh, dear. What about George Clooney? Stayed in our hotel. Yes. No. Oh, my own favourite, Super David Beckham, maybe? No.
Definitely not.
Or Mr. Daniel Craig, maybe? No? Oh, she's a hard man, hard lady to please. Well, let's find out who would be really the perfect guy for you. So what we have to do is, it's a random thing because it's a scientific experiment. So what you have to do is you have to go, he loves me, and you put it to the bottom, and then he loves me not, we throw it to one side. So he loves me goes to the bottom, he loves me not to the side, he loves me not, he loves me, he loves me not, he loves me not. So for me, it would be Super Dave, okay? But we'll see, let's see for Margie. So you have to say it as we go through. So you have to say, he loves me goes to the bottom. He loves me not, we throw that away. He loves me not. He loves me. He loves me not. He loves me. He loves me not. He loves me to the bottom. He loves me not. And the perfect guy is, let's have it. Oh! Hey! It is Alan after all. So we have scientific verification. Well done, Margie. Thank you so much. There you go. You could keep it. You could keep it. So it's true. Okay, so I hope I've been able to convince you that it really is possible to control molecular motion and make sort of molecular machine type systems. Where is this all going? Well, just as 100 years ago when they had the Model T Ford, I'm not sure they'd have predicted that Alan's car would look like this Ferrari today. And in the same way, it's just been sort of 10, 15 years since we made these sort of structures. And already we can make real motors and make molecules that move things uphill. Where's it going? I don't know, but there's many good reasons to do nanotechnology, not least of which that Jessica Alba thinks that it's great. So my thanks to Jessica, and also to my group, who are a wonderful lot from all over the world. And I'll just finish showing you some pictures of Edinburgh, which I've just moved away from, and this is where I used to live, in the new town of Edinburgh, all built in Georgian times in Great Stewart Street. And if you go down here into Ainslie Place and through into Murray Place, down here into India Street, then you come to, into India Street, you come to 14 India Street. And 14 India Street is the home of, is the birthplace of James Clerk Maxwell, who we met earlier. So James Clerk Maxwell was one of the three greatest scientists of all time, along with Newton and Einstein. Not because of Maxwell Demon, but because it was Maxwell who discovered that light was really electromagnetic radiation. And that's an achievement of which Richard Feynman, the famous Nobel laureate of physics, said 10,000 years from now, there can be little doubt that the most significant event of the 19th century will be judged as Maxwell's discovery of the laws of electrodynamics. And that's an astonishing thing to say, that the most important thing that happened in an entire century wasn't what a politician did, wasn't what a soldier did, wasn't what a president did or a general. It was what a scientist did and what a scientist thought. And you could say the same true for the 17th century, the 18th century, the 20th century. And it was great seeing so many young people, school kids from the Geneva area yesterday, because with them I think that the 21st century is in great hands as well. So thanks very much for having me here. And I'll leave you with one final thought, that sufficiently advanced technology is indistinguishable from magic. Thank you.
I'm extremely glad that your experimental proof showed that my wife was right. This is so we now have time for discussion and questions. So if you have questions, please write them on the cards that have been distributed. I would remind you that you're welcome to ask general questions on science. We've had a lot of questions the past few days, so please don't be disappointed if we cannot answer all the questions. But if you wish to leave, then please leave quietly and quickly now. Otherwise, we will carry on with the discussion. Yeah, super. think we can begin. We'll make a very bad start to begin with because there's a question directed to Dr. Roderick McKinnon, who should be landing in New York at this moment. So, but I think the question that he asked can easily be addressed by the members of the panel. Practitioners of alternative therapies often refer to subtle energy fields around human bodies. Anybody got a comment on that?
Well, I think that I'm a big advocate of evidence-based medicine. And by that I mean the current methodology for judging whether a treatment is effective or not has to do with randomized clinical trials, okay? Blind randomized clinical trials. Now, why do we need blind randomized clinical trials? The reason is that the interaction between our brain and our emotions and the rest of our body is highly intricate and very complex. There are cases where of the placebo effect, where the power of suggestion has affected a cure, that are absolutely remarkable, okay? For instance, people have done a knee surgery on two groups of patients. In one case, they did a fake surgery. That is to say, they went through the entire motion, they anesthetized the patient, etc., but they didn't actually do the knee surgery, okay? And the patient, then woke up, etc. And in the other case, they did the knee surgery. And it turned out that a remarkable number of the people on whom the fake surgery had been done actually showed an improvement, in their knees, okay? Now, this just shows you how powerful our brains are in terms of suggestion, okay? And so if you look at all these alternative therapies, nearly all of them rely on that, okay? Now, you might ask, well, what's wrong with that? Why not, use the power of suggestion? Well, it's okay if both the patient and the doctor are aware of what's going on, okay? And if they consent to it, that's one thing. The other problem is that we don't know how to harness this power of suggestion in an effective way, okay? It's highly erratic, it's highly reproducible, and it's highly variable from one patient to another. So it's not a standard protocol for treating anything. And that's why when we talk about medicines, about modern medicines, they all have to face a test. That is it due to the medicine, or is it due to the doctor telling you, here, take this, blue pill and you'll be fine, okay? And it might just be sugar, okay? So that's the objection I have to alternative therapy, which is that there's no control, and it's simply relying on the power of placebo. And that's why modern medicine is very, very rigorous and very careful to do randomized clinical trials.
Okay, now we have a question. Somebody says, well, since associated with all movement is an energy, and since energy involves a loss of mass, is the mass of molecules going towards zero with time?
No. So the molecules move, the components of the molecules move, so everything is moving at the molecular level, but that's not consuming any energy at all. So it's not the same as causing movements in the in the big world that we live in. So if I, in order to knock this bottle of water over, I have to hit it and give it some energy to fall over, and that energy then is converted to heat, which would be dispersed. But at the molecular level, everything that has a temperature, so everything warmer than absolute zero, which is only found in the outer regions of space, is actually moving anyway, and that movement doesn't consume any energy at all. So that movement of molecules doesn't cause heat to dissipate.
Okay. Question here: what are the chances of creating machines to make liquid fuels out of solar energy, from water and?
Yeah, so, well, I really believe in the future of molecular machines, not necessarily the work that we do, but the generations that will come after us, simply because biology uses molecular machines for everything, to power every aspect of life. All aspects of life depend fundamentally upon molecular machines, and biology isn't using molecular machines for no good reason. And when we're able to make molecular machines and understand how to really control motion and get the components to interact in the way that we want, we'll really be able to do amazing things such as harvest energy from the sun more efficiently, I'm sure, and also use molecules to actually make molecules which could be fuel molecules. So I really believe that those are possibilities in the future.
Another question. You're a chemist. You ended your talk talking about a physicist, and your examples were biological. And this concerns everybody. For science education, should we really have the separation of the three subjects?
Difficult one. So, but I think that we've already heard this week about how broad science is becoming and how interdisciplinary you have to be in order to tackle the big problems of today. And a lot of our work is inspired by understanding how biological machines work, and other parts of our work are based on understanding of what physicists have known for a long time and translating that into chemical terms that chemists didn't understand, that those things were known in other disciplines. But I actually believe you need to have firm foundations in the underpinning disciplines. So I don't think that it's wrong to learn chemistry as chemistry and biology as biology and physics as physics and maths as maths, because these give you these are useful frameworks of which to transfer knowledge. And it's then once you have these building blocks of knowledge to build on, then you can start understanding how to move between the others. It may be that you can do just as well by having broader learning through broader themes. But I think this compartmentalization of things at an early stage is actually quite useful. That's my view.
What do other members of the panel?
Yeah, I think, you know, a simple analogy is if you take this auditorium or this building, it has a physical structure, you know, the masonry and the beams that hold it up, etc., which are the role of a civil engineer. It has an electrical system which a civil engineer may not be very familiar with, and which has its own technical problems and its own training that's required to install it. And it has plumbing, I'm sure, which again requires a highly specialized knowledge and highly specialized tools. So now it doesn't mean that they don't talk to each other when they construct this building. And so I think, you know, David's absolutely right. Each discipline has its own language and its own knowledge and rigor, and you need to learn that before you can then crosstalk with other disciplines to create something that's bigger than just the discipline alone.
We all agree.
Okay. What would be the ideal designed molecular machine?
The ideal design of a molecular...
Ideal designed molecular machine. Well, what would be a machine that you would like to make, shall we say?
Another very good question. So the ones that I talked, really understanding what it's going to be possible to do to make with synthetic molecular machines. It's a bit like you saw how primitive the examples were that I was giving, and some of those are as sophisticated as anyone's ever made in terms of synthetic machines, and you can see how really simple they are. And so it's a bit like asking Stone Age man, just after he's made the wheel, to predict motorways and cities and so on. There's no point trying to make something that's too complicated yet. You can have visions that it would be wonderful to be able to make molecules that could self-replicate and or that could harvest energy from the sun and do it better than you can do with non-molecular machine systems yet. But I think at this stage we don't know enough about how to make the molecules interact with each other in the way that we want in order to design those sort of things. So we try and take medium-sized steps in order to get where we're going. I think we've taken the first baby steps, but it's still very early days for a field. But I'm sure that in my lifetime we're going to see some really exciting developments, not in the next 5 years, but in the next 10 and 15 years.
Thank you. Do you want to say something?
Well, I was just wondering how far away you think we are from self-replicating machines, and especially ones that can mutate.
Yeah, so I think that if those are machines that are made out of RNA or even proteins, that might be, or peptides, that might be possible to make, even with today's understanding of how things work. So those sort of things I don't think will be too far away. And I don't think it will be so far away where we make real self-replicating molecules that people can say are really have many of the characteristics of life. So artificial life, I don't think that is far away at all. It wouldn't surprise me at all to open Science tomorrow and see Craig Venter had done it. So those sorts of things could happen tomorrow, or they could be in 5 years, or they could be in 10 years, but I don't think they're going to be longer than that.
That raises the question, somebody talks the famous phrase, science sans conscience, science without conscience as meaning this. What about the people obviously are rather frightened about self-replicating molecules and afraid they'll sort of fill up the room.
Yes, but so again, so in science, it's very difficult to say that things are impossible unless they break laws of physics, and probably they need to break 2 or more laws of physics for you to be sure that they're going to be impossible. But there's different levels of science fiction. So, for example, life on other planets is something that is science fiction because we don't know that it's there, but it's actually something that many scientists are pretty sure is likely to exist. Whereas something like time travel, it would be something that would be extremely unlikely to exist, most scientists would think. So what about self-replicating machines? Are they going to make grey goo that can overtake the planet? I don't see how. I think that's at the same sort of extreme as time travel, because these things are going to have to consume a fuel. Nothing can replicate without consuming a fuel. And biology isn't able to make creatures that can consume anything, and it seems very unlikely that we're going to be able to design anything either by accident or through design that would also be able to do that. And so I think that the chances of those things happening are so infinitesimally small that you're just as likely to find someone coming back from the future and telling you that it hasn't happened.
We have a social question. Are you connected scientifically to Fraser Stoddart?
I am indeed. I am his child, not through genes, but through him being my PhD supervisor. So yes.
Where is memory located in nanomolecule machines, or is memory the wrong notion?
Yes, I'm not sure what that's. Where that's
Well, I read it.
Where's memory located in nanomachines? The structures, the molecules don't have any memory. They exist as a function of state. So if you make them in one sort of shape, they will, with the components in one position, they'll stay in that position until you move them to something else, and then they won't remember where they were before. It's the same as if you just, it doesn't remember the route of how it got there.
And I think the last question, I don't understand either. Lavoisier told us that nothing is created, nothing is lost, everything transforms. Molecular machines, are they involved in this sort of this state of affairs?
Well, from the point of view that, as I was saying, even we were talking about making molecules that can build other molecules, and I gave the example of the first sort of molecules that we're doing of being able to construct short peptides. But they have to use building blocks. They're not creating the peptides from nothing. They're using building blocks that are attached to a track, and those building blocks had to be attached to the track in order to be used as a fuel. So these no molecular machines will ever be able to create matter that wasn't there before. It's using the atoms that were in a fuel substrate and converting that into something else. Just like when you bake a cake, you take eggs and flour and sugar, and those ingredients are converted into the cake. The cake is never going to weigh more than the ingredients that went into it. And it's exactly the same when making molecules.
Okay, well, I think we've answered all the questions. You've seen the beginnings of what could be a very exciting field, and I'd like you to join me in thanking David once again.